Okay, so there are certain traits that don't follow all of the rules like typical Mendelian genetics, where it's simply just dominant over recessive. And so there needs to be times when we talk about that difference. So that's what we're going to do in this workshop is we're going to talk about um, what those differences are and when we see them. So there are three different kind of types: codominance, incomplete dominance, and multiple alleles. So first is codominance. So when you look at this cow, you can tell that it looks speckly and has kind of a reddish color and a whitish color mixed together. So hair color can be controlled by lots of different things, but specifically this kind of cow that's called a roan cow, where it has a mixture of these red and white hair, is an example of codominance. So this is when the phenotype of the heterozygous individual expresses the phenotypes of two dominant traits. So, what I mean by that is that um, we are going to be looking at that heterozygous individual greatly. So, sorry, this keeps acting goofy. Um, so, what I mean by that is, if we have, say, an individual that has this genotype, it has um, C, C, and then we have an individual that is W, W, then the heterozygous would be like a CW. This heterozygous individual is going to be considered codominant when it is going to be a combination of both of these individuals. So a third new phenotype is expressed. So both of the phenotypes from the parents are going to coexist in the same organism. And we usually use capital letters to represent this. We don't use capital and lowercase because that implies dominance. So a good example of this is the red, white, or roan color that I was talking about. So if you had big R is for red, big W is for white, the RW, the heterozygote, would be this roan color because it's coexisting on the same organism. Now, another example is incomplete dominance that doesn't follow Mendelian genetics. So this is where the individual's phenotype is a blending of two colors. So pink flower color in a lot of flowers is not something that's standard or something that is produced simply by dominance. It's produced by incomplete dominance because it's a blending between this red color and this white color. Okay, so we also use capital letters for this. As you can see in red snapdragons, capital R is for red, white, capital W, and then the pink is going to be a heterozygous between the red and the white. So you'll see two different letters there. So I always tell you in examples if we're dealing with incomplete or codominance so that you know, but you should also know whether it's blending or just a mixture of the two to know whether that's incomplete dominance or codominance. And so this one is where one is not completely dominant over the other as well. So the third type that does not follow the typical rules is multiple alleles. So this is something like hair color. We don't just see one color of blonde or one color of black. We see this gradient. Okay, so as I said, this is going to be where you have more than two alleles that are producing a specific phenotype. So you're going to have multiple alleles doing that, and it's a range of things. So let's look at a few examples here so you can identify the different types. So first, let's look at this one. Is it codominance or incomplete dominance? So we have a black chicken here, we have a white chicken here, and then we have this kind of speckledy chicken. So the genotype for the black one is big B, big B. Genotype for the white one is big W, and the speckled one is big B, big W. You notice, like I said, we use all capital letters. So since this is a mixture of the two, and you see both of the phenotypes of the parents, this one is codominance because the two phenotypes are coexisting in the offspring. So then you'll see the difference on this next one, where we use the same types of letters for the genotype, but the offspring down here is a blending of the two parents. And so when you see a blending, that is going to be incomplete dominance because this red is not completely dominant over the white. Okay, so then on this next one, we're going to actually try an example. So this one, it says the trait for feather color in chickens follows the rules for codominance. So remember, that's a mixture of the two. So make sure you pay attention to that. And illusion is a combination of black and white feathers. So for the first one, which we're going to do right here, a black feathered chicken is crossed with a white chicken. Okay, so just like before, you follow the rules of dominance. So we know black is going to be dominant to white. So you have a black chicken. So big B is crossed with a white chicken. Okay. So since we know that they are following codominance, they're both going to be capital letters. Okay. So then when we combine our letters in the box is, sorry, my handwriting kind of stinks on this. Okay, what percentage of their offspring will be Andalusian? So the answer to that is 100%. Okay, so all of them would be Andalusian. Okay, so then for the second one, it says 2 Andalusian chickens mate. So we know Andalusian is this phenotype, or this genotype, I'm sorry. So if we cross those two, we're crossing 2 that are BW. So now when we fill out the Punnett square, we're going to get kind of a different ratio for the offspring. Okay, what percentage of their offspring will be black feathered? Okay, so black is big B, so that's 1. So that's 25%. Okay, and then Andalusian are these two. So that's going to be 50%. And then their white is going to be 25% again. Okay. So it's kind of nice because you just assign letters and you know they're going to be capital and then just fill out your Punnett square like normal. Okay, so let's try another one here on the next slide. But this one is going to be incomplete dominance. So this one says that snapdragons follow the rule of incomplete dominance. So with that, you know you're going to use capital letters. And a red snapdragon is crossed with a white snapdragon. So red is going to be capital R, white, capital W, capital W. So we have R, W, R, W. Okay, so then what are the possible genotypes of their offspring? So obviously, like all of them are all W. And then the phenotypes, they're all going to be a blending of those two colors, right? And so a blending of those two colors is going to be pink. Okay, so then if we do a second one, red is crossed with pink. So then here, it's going to be kind of like the last one. We're going to have 50% red and we're going to with 50% white. Or I'm sorry, 50% red, 50% pink. Okay, so the last thing we're going to talk about with this is blood types. So blood typing is something that uses a couple of different ones of these that we've just been talking about. So blood typing is going to use multiple alleles and codominance. And so the thing that's interesting about blood types is that even though there are 4 types, they all are being used in different times within the population. So, that's why it uses multiple alleles. It's going to be expressed within the population. It changes based on how it's inherited and how many people have it at a particular time. So there are 4 blood types. There's type A, type B, type AB, and type O. The reason it utilizes codominance is because blood type AB is meaning that there are proteins that are expressed for each of these. There's a type A protein, there's a type B protein. type AB expresses both of them at the same time on the surface of the red blood cell. So that's why it's called codominance, because they coexist at the same time. So you inherit your blood type from your parents, and I'm curious, how many of you actually know your blood type? So message me if you do. I think it's interesting. Hopefully you've donated blood, and I know you probably haven't, you're too young, but hopefully at some point in your life you will donate blood. It's a good thing to do for sure. Okay, so then let's talk about how blood type is inherited. So the genotype for blood types, if you're type A, you can either be like AA or you can be AO. So type O, you have no proteins on your surface of your red blood cell. It is just completely plain red blood cell. And so that's why they use like these little eyes down here. Now, if you are type AO, then they represent the genotype by I, big A, and then a little I. Type B, you can be the same way. You can be BB or you can be BO. And then type AB blood, you have proteins for both A and B. So you're probably thinking like, this doesn't make any sense. So let me show you this table that will make a little bit more sense. So look at these pictures in this first row. Type O blood, as you can see, does not have any proteins on the surface. Group AB has both types of the B that you see here and the A that you see here. So within our blood, we have antigens in our bloodstream that are basically just different types of proteins, and they're antigens that allow us to recognize our blood cells that we have. And so that's why it's important that we get the right blood type when we get a blood transfusion. Okay, so we have A antigens in our blood, and so when they do blood typing, that uses anti-A serum, and so it recognizes these A's to tell us that we have type A blood. For type B blood, it's B antigens. For AB, it's both, and then for O, it's none. And so then we also have antibodies in our blood. So antibodies are produced by our white blood cells, and it helps our body to recognize things and either attack them or recognize them as part of our body. So type A blood has anti-B antibodies. So that if you were given, say, type B blood, it would attack it, and white blood cells would attack that type B blood and kill it because it doesn't recognize it as part of your body. So type B would have anti-A, and this is because it would recognize type A and would kill those blood cells. Now, AB doesn't have any because you can have both types of blood, and you can be given either type because you have both proteins. Now, group O, you have anti-B and anti-A because they can't be given either because your body would reject it because they didn't have these proteins on the surface. So this is important for when you do the activity you're going to be doing with this, and it's a virtual blood typing lab. And then just to kind of look at like how can people donate and receive blood. Type O blood is considered the universal donor because it does not have those proteins on the surface. It can donate to anyone. The most restrictive type is type AB for a donor because they can only donate to type AB. But you can see A can donate to both A and AB, and B can donate to both B and AB. So not only do you have to worry about having the correct blood type when you are donating blood or receiving blood, you also have to worry about something called the rhesus or Rh factor. So some of you might have heard about this with pregnancy, that up here on the right-hand side, it's called the rhesus conflict. So the Rh factor is another protein that's inherited on the surface of your red blood cells. So if you are positive, then you have the Rh plus, or you have that protein. If you're negative, then you don't have the protein. So my blood type is A positive, which everybody likes to tease me. Oh, it's A plus, because I was an overachiever as a student. So people like to tease me about that. And so if you are pregnant and your child has a different Rh factor than you, so say you're negative and your child is positive, then that can cause problems during the pregnancy. So if that does happen, you can get a blood test. You always get a blood test early on in pregnancy. And if you're different than your baby, they give you something called Rogam, and it's just an injection. And they give you that injection usually early on in the pregnancy and periodically throughout the pregnancy, and then really close up to the time of giving birth, just because you're going to be losing a lot of blood then, and they want to make sure that there's not going to be any issues at that time. And so you also have to consider that when doing blood type. So this big table I'm going to show you next is showing all the different blood cell compatibilities when you include the positive and negative as well. So notice if the donor has a positive, they can't donate to any negative. So see negative, no, 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 no. You'll notice that AB positive, you want to be AB positive because you can get blood from anybody, as you can see here. So that's awesome. And as a donor, you want to be O negative because you can donate to anybody as well. So this is just kind of an interesting thing. I think blood typing and blood donation is very interesting. So like I said, once you get older, be sure to do that. It's a good thing. The Red Cross always does lots of blood drives throughout the year. Okay, so I think that's it for this workshop.